The extraction of DNA of a good quality is important for the study the molecular genetics of an organism. The genetic material is in the nucleus of the cell and in some organelles, like mitochondria, chloroplast, and ribosomes. It is important to know the nature of the cell to choose an adequate protocol of isolation. The steps are as follows are cell lysis, elimination of proteins, and rush the concentration of nucleic acid. The most important difference between the method of extraction DNA and RNA are the reagents used to eliminate the proteins. In the isolation of RNA, must proceed carefully to avoid contaminating ribonucleases degradation, either by the same experimental material or by the operation figures. In the preparation of DNA degradation by deoxyribonucleases, they are activated by the valent metal ions. It can be avoided in the presence of gelatin agents and SDS. Another difference is that the precipitation of DNA used ethanol, and in the precipitation of RNA used isopropanol. The main difference of the extraction of DNA in different organisms like bacteria, plants, and humans is cell lysis. A cell wall and cell membrane of each of these species is different. Animal cells have membrane which consists in phospholipids is necessary to degrade and the use of detergents. To degrade the cell wall of bacteria, it is necessary to use enzymes because wall peptidoglycan not be degraded with detergents. Finally, the plants have to degrade two cell walls. It is necessary to use enzymes. In the next chapter, we have the different protocols for lactobacillus, Arabidopsis thaliana, and saliva. The main foundation that are used to in various purification methods are based on five conditions. Lysis, cells are broken. Binding, adding a buffer solution to the sample and the nucleic acid can bind to the silica gel membrane in the column. Washing, the flow through is eliminated. Dry the DNA pellet. Elution, the nucleic acid is collected for the bottom of the column. There are different methods to quantify nucleic acids. Spectrophotometry, we can confirm that sufficient amount of nucleic acids by measure absorbance in spectrophotometry. Reading writing of 0.1 to 1 and absorbance of 1 unit to 216 nanometers correspond to 50 milligrams of DNA per milliliter. Fluorimetry using fluorescence probes quantify highly specific of DNA, RNA, or proteins. Slot blood, common procedure specific to primates, have been modified using chemiluminescence rather than radioactivity. You can detect less of nanograms, can detect DNA double and single strand. Running in agarose gel, for example, in which it is known that the DNA concentration will be very low. This method will measure bromide etidine fluorescence intensity to estimate DNA concentration.
Here are the results. The following figure shows a photograph of an electrophoresis gel. It's of 0.8% of agarose. On each well, we loaded samples of 5 microliters. On the first lane, we can see a 1 kilobase enriched gene marker. On the second one is a genomic DNA sample. And the third one is E. coli RNA sample. On the figure 2, we can see that there's a DNA band on the lane 2. It's circled with red. Then on the lane 3, on upper side of the wall, we can see that the RNA sample was charted incorrectly, so that it go out of the wall. Then on the yellow box, we can see a protein contamination. Then on the green circle, we can see that the RNA also went outside the well. And on the pink box, we can see a salt contamination. On the blue box, we can see the RNA degraded, and also the sample run more than the marker. And on the also in the third lane, on the red circle, we can see that the DNA contaminated. There was a DNA contamination in the RNA sample. The following table shows a summary of the UV quantitation of nucleic acids. On the first column, you can see the sample, the type of the sample. The second column is comprised of the optical density at 260 nanometers of wavelength. The third one is the optical density of 280, and the third one is the ratio between the optical densities, and the final is the concentration in micrograms per milliliter. Then we did calculations which are based on the formula. For DNA concentration, there's a factor with about 50 micrograms per milliliter per the optical density and the dilution factor. We get a DNA concentration of 735 micrograms per milliliter and an RNA concentration. To calculate this, we first change the factor, which is 40 micrograms per milliliter, and also the
Comparing both protocols, the main differences between procedures lie in incubation periods, spin speeds, and amount of centrifugations, the use of different extraction buffers, different precipitation substances, among other things. The difference on the protocols are because of the complexity of cells from where we are obtaining DNA RNA samples, also because of different molecular weights and interactions within molecules and the medium, the presence of DNA digestive enzymes or RNA digestive enzymes, and therefore the different inhibitors for each. According to the Institute of Biotechnology of UNAM and the National Institute of Ecology in Mexico, to consider a sample of DNA pure, the absorbance range must be above 1.8. Below this mark, there must be protein contamination. Ours was of 1.5139, but according to the electrophoresis gel results, no protein contamination was observed on well 2. So we attribute this absorbance to the fact that our DNA sample was quite diluted with water from molecular biology. About the RNA sample, the absorbance obtained was of 1.3449, and the protocols remark that a reading above 2 can be considered a pure sample of RNA. The above, in addition to the electrophoresis results, we can conclude that our RNA sample is in fact not pure, as it can be observed on the gel on well number 3 that there is an excess of salts, a contributing factor for impurities, a probable contamination by DNA sample, and a degradation may be due to no inactivation of RNA digestive enzymes. All of the above must be taken into consideration when performing a PCR, sequencing, or an enzymatic restriction, because they can be inhibitors and contributing factors that can alter results by increased concentration of contaminants.
The most important points to consider in the isolation of DNA is the cell lysis, as already mentioned. It is necessary to know the nature of the cell to identify which protocol to use and obtain DNA in good conditions. From quantification of nucleic acid through spectrophotometry, it is important to look wide and should be done with the same substance which the DNA was resuspended. It is also necessary to make the necessary dilutions for an accurate reading.